So, here we are. So, the topic of my talk today is niche evolution along environmental gradients. And that's, of course, something relevant to the problem of adapting in a changing world with in particular, in the context of global change. So I will present you, it's about experiments I've been doing in my lab, different experiments. So when you are trying to study niche limits and shifts, you got, of course, to find the traits responsible. There are many talks talking about this, and possibly plastic traits, and the genes, and you got to understand the ecology of that, and that's all the 3 steps are fairly difficult to do. And a key issue with respect to global change is to understand niche evolution when you've got an abrupt environmental change compared to generation time. And in my talk, I will use example from extremophile organisms that have adaptation to kind of extreme environmental abiotic factors. So, so they live in this kind of very extreme environment in terms of abiotic temperature, pH, salinity. And so I will tell you about 3 stories. The first is about adaptation of bacteria, and the environmental gradient is pH. The second one is adaptation of artemia, so a small crustacean, in hypersaline environment, and it will be about adaptation to temperature. And the last story is about both artemia and bacteria together along non-environmental gradient, which is salinity. So, before I start with these 3 stories, I'd like to highlight a few important points that I will illustrate with these experiments. Is that there are many issues when you want to understand the adaptation along a gradient. And here I want to highlight 2 of them that I find particularly problematic. The first one is that when you want to study niche evolution, you are doing some measure of performance across a gradient. And often measuring performance and measuring fitness is not always very clear what you are doing. So I will elaborate on this a little bit. And in particular, there is this issue of habitat quality. So I will explain in a minute. And the second issue is that if you want to understand how the niche changes through adaptation, you got to have a way to measure niche deformation, whether or not it's very rigid or it can move kind of freely. So that's the problem of understanding the origin of trade-offs and the role of plasticity, evolution. So I explain a little bit what I mean with this habitat quality issue. So if you take a cactus that have been adapting for a long time in the desert, and you bring it in the greenhouse, and you try to have it growing. So imagine that this quantity of water is the quantity of water you'd usually have in the desert, the place where the cactus have been evolving for a long time. And you take as a measure of performance growth. Then if you give them more water, you will have more larger growth. And of course, if you give them too much water, then they will die, of course. But the place on the gradient where you got the highest performance is not the place where these plants are the most adapted to. So there is a mismatch between the history of adaptation and the performance that you can measure. And that's, of course, due to the fact that you got different habitat quality. More resources here, more water means higher metabolism, and you can have higher growth. And this phenomenon is quite general. It's not only due to this particular example. You can take bacteria and you grow them. For instance, you take E. coli. It has been evolving at 37 degrees in the gut. If you try to grow E. coli in the lab at slightly higher temperature, you will find that they grow a little bit faster. It's the same. And of course, if the temperature is too high, then they don't grow anymore. But the place where you've got the optimal growth is not the environment where these creatures have been evolving for a long time. There is a mismatch, and it is due to probably to something like we can call habitat quality. So, if you want to write that the measure of performance is some kind of a product of two things. One is adaptation, the fact that the traits have been evolving in those particular organisms to match some optimal value, and the fact that the different environments are different in quality. Then you've got, you don't have a direct relation between the performance and adaptation. And imagine now that you are trying to investigate the adaptation, the niche deformation of a particular species. So you got the two red curves here. One is dashed line, and imagine that you got an increase in due, for instance, to plasticity in niche width. But that in parallel to this, you've got an habitat quality, which is in blue here, and that you measure performance. What you will see here is that you've got a shift in the niche, but the shift in the optimal value of the niche is not due to a shift in the underlying adaptive trait, where you just have a change in width. So when you're trying to analyze a deformation of niche, you got this problem of disentangling what is due to habitat quality and what is due to adaptation. And of course, you got on top of this a problem of having possible plasticity in the traits. So if you've got a trait optimum that is along an environmental gradient here, you got a particular value for the trait. Here it's not a plastic trait, so you will have some kind of measure of fitness in red. And if you've got a plastic trait, so you've got a reaction norm, you've got a variation of the trait with the environment, which, for instance, in that particular case, leads to a larger niche. So you can envision different scenarios for niche deformation. The first thing is that, so imagine you got a new environment. So here it's a pH gradient. You will see in a minute that I'm doing experiment with pH on E. coli. So you've got an old environment, and then you transfer them to a new environment, and the trait may change very locally in this new pH condition. And if you got such a local deformation of the reaction norm, then you will have a corresponding change in fitness that would be illustrated in red here, in 1 or 2 and 3 steps along the process of adaptation. And if you measure relative fitness of the evolved line against ancestor, you will have just a change in relative fitness around this new condition. If you've got a global deformation, and that's most models right now use this kind of way of thinking, where you got when you change, so you got a change into the new environment, and what is changing is the intercept of the trait to match the new optimum. So in 1 step, 2 steps, 3 steps, you match the new optimum, and so that corresponds to a sliding niche model where the niche slightly optimum move to the new optimum. If you measure relative fitness, then you will see something like that across time. You can have also models of global deformation where you got the evolution of plasticity in the same time, and talks have been mentioning this possibility yesterday. So in particular, you got this what I call this sidestep niche model, where to adapt to a new environment, you've got first an increase in plasticity. So at step 2, you've got the trait which becomes more plastic, and then the intercept change. So you've got the niche which becomes wider, and then it shifts. So that's in particular has been modeled in a Landy recent paper. And so when you measure relative fitness, you will see this pattern through time. So now I will switch to my 3 stories. The first one is experimental evolution in the lab to try to investigate these niche deformation models. So it's about E. coli and a pH gradient. So the idea was to to have, so these bacteria are adapted to this kind of average pH around 6 or 7, and then you transfer them to a very acid pH. So in this case, in this experiment, it's 5.3, and you let them evolve for a long time. So we mark the bacteria with fluorescent proteins so that we can distinguish them and measure fitness. And they lose the fluorescent protein across in extreme pH, so we investigate this range. So it's a very classic experimental evolution design where you got evolutionary replicates, here 4, and you replicate them through generation. Here it's 2000 generation at a new pH condition. It's an abrupt environmental change, and you want to see what the niche deformation looks like. So you measure fitness by taking samples at any point in time and measure the relative fitness against the ancestral strain in different pH conditions. So it's a massive experiment where you've got 1,000 competition and a huge, through flow cytometry, so you can get very precise estimates of fitness variation. And to remove the effect of the marker, you can, I won't go into details, but you can remove the effect of the marker, the fluorescent protein, by performing different competition between the different genotypes. So here are the results for directly the effects of the relative fitness against the ancestor across a pH gradient, which is from 5 to 8.3. And that's the 4 evolutionary replicates. So for instance, in the green one, the first one, what you see at 500 generation, 1,000 and 2,000. And so you've got an increase in relative fitness in an acid condition and a decrease in a very alkaline condition. And what you can see is that 500 generation, you don't, the fitness here, the relative fitness has not been reduced in this particular case. And you got the same in this replicate. And here you got a drop in fitness in this range. You can notice that there is very little fitness variation around these pH values in this range. And you'll see why just later. So if you want to analyze this kind of data in terms of niche deformation, what you can do is you take a niche template with a center and some shape, and then you can have a deformation. So for instance, you can have a right stretch of this niche template so that you get something like that. Similarly, you can have a left stretch, and you can have a vertical shift across all pH conditions. And so you can get, so you got to choose some fairly simple niche template, and you fit this to fit relative fitness data to avoid the problem of habitat quality variation I was mentioning at the start of my talk. So here is the result of this analysis on this data set. So the different colors represent the different evolutionary replicates, the 4 of them. So that's the deformation parameters, but you can focus on the width of the niche illustrated here and the center of the niche illustrated here. So you see that the center shift, that's a minus here. So the center of the niche shifts to more acid condition as you expect, because you select in 5.3. But you can see that in 2 replicates you've got a transient increase in niche width early on, and then the niche width decrease again, which is exactly what we saw before. Oops, sorry. Well, because here you don't have any drop in fitness in this 8.3 pH. So what it tells you is that you've got, so that's the fitness niche template that is fitted with this data, where you got a very large plateau around these pH values, which mirrors the range of pH you get in mammal's gut. And in this range of pH, you don't have any trade-off in the fitness, so that fits the fact that you don't see any relative fitness variation when you shift this niche in this range. So I skip this thing up. So the one key question is whether this very simple and global deformation model capture sufficiently well the data. So here is the correlation between the observed selection coefficient and the one which is fitted by this deformation method. And you can see that you almost explain all the variation with a very simple deformation model. And so the conclusion from this is that you've got a strong support for this global deformation of niche when you got evolution in a new environment, and that you don't have only this sliding niche scenario. You got this transient possible sidestep scenario where you've got a transient increase in niche width. So my second story is about niche evolution in the wild. So you take it was in the lab. Now you want to go in the wild to see what you can do. So here the idea is to use Artemia, so this little crustacean, to measure adaptation to temperature. So these creatures are so a little bit of background. They are very useful for long-term studies because you got very large population sizes in the field. They produce cysts, so these eggs in very large quantities, that you can have this kind of collection of cysts that you can reuse again in the lab. And so you can use this to do population comparisons through time. And in the lab, you can do crosses. You've got the large fecundity and short generation time. And they are, so they live in salterns, for instance. You've got here many population of Artemia near Doniana. And so this is quite of a simplified ecosystem where you've got few biotic interactions. And because they are in an extreme salty salt condition, they are adapted to very strong salt concentration. So we investigated a particular field situation, which is a kind of a field global warming experiment. So in 1985, there was in Vietnam, there was a new saltern that was created de novo. And Artemia were introduced intentionally from San Francisco into this new saltern. And so you've got an abrupt change in climate, because you move from 22 degrees on average to 30 degrees tropical. And so we've got cyst collection from the start of this field transfer so that we can rehatch and study. And that's plus 8 degrees. That's twice the worst scenario of a global change that we know, that we have prediction for 2100. And so we did a big experiment where here we've got the different samples for different years of the Vietnam populations. And for comparison, we've got some San Francisco population. So you've got all those, it's about more than 100 generations of evolution for those shrimps. So we did a full SIP design where we made each of males from each of those populations that we rehatch against 2 different reference populations to avoid maternal effect and to measure whether or not these populations have been evolving through this higher temperature. So we did several measures across several temperature treatments. So here is the result of the fitted longevity across temperatures. That's the range of temperature you find in San Francisco or in Vietnam, in Vinh Châu. And so here in red, you've got the recent Vietnamese population, and in blue, you've got the old ones. And so what you can see is that there is very limited increase in longevity at the new temperature, but you've got a massive decrease in longevity in colder temperature. So here again you can see that there is this habitat quality issue because you see that longevity is much higher in a cold environment. It's just because the metabolism of those creatures is slower. And so if you want to take a comparison across environment of, let's say, longevity or survival, the same thing, then it's difficult to compare across environment what it means in terms of performance. But nevertheless, within each environment, the relative comparisons make sense. So what you can see here is that the main result is that you've got maladaptation. They lose the ability to survive in cold environment. But that might be related to the fact that in a hotter place, they need to develop at a faster rate, and that you've got a trade-off among traits that we don't capture in the fitness measure we are using here. So there is very limited adaptation to higher temperature, but there is a large drop in survival at low temperature. So the first problem I want to highlight here, despite the fact that it's a very ideal situation to detect adaptation to warmer conditions, is that there is this problem of measure of fitness. You've got a measure of survival, but it trades off perhaps with developmental rate and possibly reproduction. And we need to have some way to integrate those life history traits in a single fitness measure, which is not obvious to choose in the first place. And I think this is a very serious problem for this kind of field ecology work. And there is another problem, the fact that we don't know exactly who is adapting, but that I will come back on this on my third story. Just to advertise the fact that to analyze this data, we are developing a likelihood model called livelihood, where we try precisely to integrate different life history traits to obtain integrated fitness measure. So it's a beta test phase of the model and so of the program. So if you got data where you got full life histories and you want to do some fitness estimate, you can talk with me later. So last story is about a mixture of Artemia and E. coli adapting along salinity gradients. There is, with these shrimps, a paradox that when you investigate their distribution along salinity gradients, you see that you don't find them at low salinity. Most people think it's because you got fish at low salinity that eat them, but in fact, when you raise them in the lab, they do not perform very well either in those conditions. So why is it that they perform not very well at those conditions? And I got to give you a little bit of background for the salinity problem in those shrimps. So they feed, so in the gut, you've got the same salinity than in the environment. But within the body, you've got a very low salinity. In fact, they are freshwater, derived from freshwater crustaceans, so that they have they are freshwater emollient. So they've got pumps to excrete salts, and a priori you would think that if you put them in fresh water, they would have less work to excrete salt, and it would be easier for them to survive in those environments. So there is a paradox. Why is it that they don't do well at low salinity? And they eat these algae that you can find in salterns, the unicellular algae. And you know that many herbivores have gut microbiota to digest cellulose. And so we thought perhaps there is something about those microbiota limiting their niche. And so we did an experiment. First, we took some sample of their gut microbiota and grow them in the lab. And what you find is that that's a growth curve. Optical density of the bacteria grows through time at different salinity, and you effectively see that at low salinity, 40 or 20 or even 0, the fresh water, the gut microbiota do not grow in the lab. So to investigate this, we did a big experiment where we got 3 treatments. You got the low and high salinity treatments. Then you've got algae and yeast as a source of food, so that because probably they need bacteria to digest algae but not yeast. There is no cellulose in yeast. And then the last thing is that you've got sterile and non-sterile. We produce axenic shrimps, that is, they don't have any bacteria, so that we can remove the effect of the microflora, the microbiota. So when you look at this, you find first that, as we know before, at 80 gram per liter, they perform well when they are not sterile, and less well at 5 gram when it's more fresh water. When you remove the microbiota, when it's sterile condition, they perform not very well. And when you give them yeast, so the level of yeast and algae are not directly comparable. You got to compare them. It's not the same food source, but with yeast, they perform equally in all treatments. That is, it's not a problem of the Artemia, the fact that they don't survive well at low salinity. It's a problem of their microbiota. So it's a problem for the gut bacteria to grow at low salinity, and that's why the Artemia do not digest well at low salinity, and that's why they don't. That limits their niche. So you could imagine that the Artemia niche is not under the direct control of Artemia genes, at least at those salinities, and that even in the lab experiment, you got this ecological problem of biotic interactions, and it's difficult to know in this kind of superorganisms who is evolving exactly under which condition. And perhaps that's a confounding effect in our previous experiment in Vietnam, where we did not control for any microbiota effect. So my overall conclusion of these three stories are as follows. First, there are several problems of measuring niche deformation and maladaptation across environmental gradients. And there is this issue of habitat quality, and I've been showing how you can a little bit make some progress on this in experimental evolution. We've got evidence for different models of evolution of niche with a sliding niche and sidestep niche model. We've got data consistent with both. We've got data indicating that you've got strongly nonlinear reaction norms and fairly global niche deformation when you analyze them. Obviously, there is a power issue. That is, when you do experimental evolution with 2,000 generation and very precise fitness measure, you can say many things. And even in the best field situation I've been talking about, it's about 100 generation, it starts becoming much more difficult to detect adaptation. And this is in part due to the way we measure fitness and the methods we have, but also just the fact that you've got less adaptation in less generation. And finally, maladaptation to abiotic gradients may not depend on the genome of the focal species you are looking at, and that's something we should certainly think a little bit more when we are doing different kind of experiments on various organisms. And I think just have to acknowledge the different people with main authors on those three experiments: Romain Gallet, Nicolas Rode, and Audra Nouquet, many other collaborators and funding. Thank you.